I mean, this is excellent. So this is always a great pleasure. I'm going to talk about the work that we've been doing for quite some time about the motor adaptation without cerebellar signals. And I would like first to thank the people, or to give the credit to the people that actually did the work and the funding agencies. And so this is a joint project together with people from my lab and also several other PIs, including Firas Mawase and Jonathan Kadmon and Mana RM. But of all these people, I have to emphasize the students that actually did the work, and that is Sharon Israeli, who is a postdoc in the lab, Nir Vixina, and Hugo Nino that joined later and helped him with the data analysis. So my lab is actually interested in the way the brain and the motor system control and coordinate voluntary movements. And this is, obviously, it's a complicated problem. And one of the reasons is that there are many different centers in the brain that contribute to this ability, to our ability to generate well-coordinated and rapid voluntary movements. But of all these centers, we think the cerebellum is, of course, playing an important role. And the reason we think so is based on studies from patients, and this is the evidence, accumulated along the years, starting from the seminal work of Gordon Holmes that shows the motor symptoms of soldiers that suffered acute cerebellar injuries due to gunshot. And he showed that these patients suffer from uncoordinated movement. He actually listed a long list of impairments, including motor timing, problems with timing and coordination. More recent studies actually showed, using modern tools, study cerebellar patients, and they showed that these patients suffer from poor coordination, inappropriate timing of movements, but also impaired adaptation to external perturbation. So the questions we asked is how the cerebellum, that is located a little bit far at the back of the brain, affect motor output and motor control in humans and non-human primates. So the cerebellum has access both to cortical and subcortical motor structures, including the motor cortex and the spinal cord. But with evolution, these efferent pathways of the cerebellum actually change their output or change the balance in connectivity, such that in humans and non-human primates, the most direct or the most important cerebellar involvement with motor control is mediated via the ascending pathways from the from the deep cerebellar nucleus via the motor thalamus to the motor cortex. So this pathway actually became much larger on the expense of the descending pathway, which is the cerebellar rubrospinal pathway. So now what we ask is, what are the cerebellar to motor cortex interactions which underlie motor control and specifically motor adaptation? Now, in order to study motor adaptation, we actually used a very common approach. We trained monkey to perform a center-out reaching task, and the monkey is wearing an exoskeleton, a kinome system, and the task includes both a pre-cue period, delay, and reaching movement. And during task performance, the monkey needs to overcome force field, velocity-dependent force field, curl field. And we are studying how the monkey is actually adapting to these perturbations. We measure the trajectory of the monkey. We measure the deviation of this trajectory from the straight line. And we use this mirror, this deviation, this errors measure as a way to quantify the adaptation. And we see that along the trial, there is a decrease in the error, showing that the monkey can adapt to this external perturbation. But in order to understand how the cerebellum is actually involved in this process, we need some way to perturb the system. So what we are doing is we are relying on a method that was developed in our lab, where we implant we implant a chronic stimulating electrode at the superior cerebellar peduncle, which is basically the afferent pathway of the cerebellum, and we use single pulse stimulation in the peduncle in order to activate the cerebellar thalamocortical pathways. And using this single pulse stimulation, we can also identify neurons that are part of these pathways, both in the motor thalamus and the motor cortex. But we can also apply high frequency stimulation, or in short, HFS. And we found that this HFS actually prevents the flow of information in the cerebellar thalamocortical pathways and produce an effective block of information flow in these pathways. And we know this perturbation is effective based on the behavior. We can see that under HFS, the movement becomes dysmetric. You can see here, this is trajectories the monkey performed under HFS compared to control trials. Timing of movement is affected, reaction time increase, movement time, velocity decreases, the curvature of the movement becomes larger, and there is a discoordination or a disintegration of the multi-joint movement. In addition to the behavioral impairment that we found, we also found that a cortical response to stimulation is abolished under this protocol, under high-frequency stimulation, and cortical activity changes in a manner that actually replicates the finding of people that studied the cortical activity under dentate cooling. So when we cool and abolish activity in the deep cerebellar nuclei, we get very similar changes in motor cortical activity as we found under this high-frequency stimulation. So from this point on, I will use HFS as a way to reversibly and effectively inactivate cerebellar flow or cerebellar outflow of information. The cerebellar signals are blocked. So now we are studying the effect of cerebellum on a force field adaptation by implementing this not only force field, but also high-frequency stimulation. So we trained the monkey, as I said before, on this center outreaching paradigm. And in a single session, the monkey actually performed several types of trials. So every single session starts with a control 8-target task. The monkey actually make movement, unloaded movements to one of 8 targets, as shown here. Then after this set of trials to one of the 8 targets, we select the learned target. And from this point on, the monkey will perform a set of trials all directed to the learned target. But during these trials, we will implement different type of perturbation, which includes a force field, applying force field, applying HFS, or applying force field and HFS. And between these different perturbations, we have washout trials that actually separate these manipulations. And this set of trials allow us to actually study what is the effect of the loss of cerebellar signals on motor adaptation and motor cortical activity. So the first part, I'll try to describe what happens to force field adaptation under high-frequency stimulation when a cerebellar signal are lost or blocked, reversibly blocked. So this is just an example from a single session. You can see here the movement the monkey is making towards a single target, the learned target, under control conditions and under force field condition. And of course, the force field condition, the monkey now needs to overcome this velocity-dependent force field. And you can see that there is a response. First, I mean, the deviation becomes larger, and gradually the deviation from the straight line becomes smaller and smaller, showing that the monkey is able to adapt, at least to some extent, to this force field. Now, when we use the same protocol, but in addition to high-frequency stimulation, we can see that the trajectories changes. And specifically, we have high-frequency stimulation actually increases. This is high-frequency stimulation alone, increases the somewhat the variability of the performance of the movements. However, combining high-frequency stimulation with force field actually increases the noise in the performed trajectories even further. And we can see this when we quantify, when we measure the deviation or the error from the straight line as a function of trial number. And we can measure these deviation either at early point in 150 millisecond or the maximal deviation. And we can see that in both cases, the monkey cannot adapt well to force field under HFS. And the red curve actually shows this, and you can see that the residual deviation or the residual error is significantly increased under HFS compared to control trial. So adaptation is impaired under HFS. However, we found that this change, this is not the only change in adaptation when cerebellar signals are lost. And another finding that we have is that the adaptation process is not only incomplete, but also noisy. And what do I mean by noisy? When we look at the average profile of adaptation, we can see that there is a gradual decrease of error with trials. So the adaptation is actually reveal this inverse correlation or negative correlation between the size of the error and the trial number. And if we measure this correlation coefficient between the size of the error and the trial number, we can see that we have a clear negative correlation between these two values. Now, this actually shows us there is a monotonic decrease of error with adaptation. Now, this is the average curve, but also when we take a single session curve, and this is just the errors the monkey performed in a single session, the curve is much noisier, but still we have a negative correlation of, in this case, minus 0.57 between the magnitude of the error and the trial number. This negative correlation is reduced under HFS. Here in this example, it's only minus 0.18. And in general, what we found is that the correlation coefficient or the negative correlation, which is corresponds to this monotonic decrease in error, is weaker under HFS compared to control. Now, this is when we measure the deviation in the first 150 milliseconds, or when we measure the maximal deviation, we can see that there is a significant change or loss of this monotonic decrease in error under HFS. So monkeys not only are impaired in their ability to correct for the error, the entire process is noisier. And finally, we use catch trials in order to quantify the adaptation process. And here you see just two examples of the movement the monkey is making under force field and the catch trials in color, under control force field and force field HFS. These are just two examples. And using these catch trials, we can actually measure what is known as the learning index. So this is just the definition of the learning index taken from Chen et al. From 2006. And here we find that also the learning index that is calculated based on the catch trials is significantly lower under HFS compared to control trials. So all these measures actually shows us that adaptation is significantly impaired when cerebellar signals are lost. Next, we try to estimate what are the mechanism. What is how HFS affect the adaptation process? So here we use the framework that was shown very nicely at Albert et al. And I mean, it's kind of was suggested even earlier than that, but this was just a very nice figure to explain this. And according to this model, adaptation is actually represents a balance between two factors, the error sensitivity and the retention factor. And so we computed these two parameters, the retention factors and the error sensitivity, under control and HFS for the data that the behavioral data that was collected in our experiment. And what we found is that error sensitivity is significantly decreased in the presence of HFS compared to control trials. Retention factor is also significant decrease, but this change was much weaker. So the most dominant change that we found under HFS was a decrease in error sensitivity. So, but there is some problems, because in this study of Scott, Albert, and Reza here, they also showed that error sensitivity is in itself sensitive to external noise. So in the presence of noise in the external perturbation, error sensitivity decreases. So I showed you in previous examples that under HFS, we have enhanced noise, that there is enhanced motor noise. Performance is much more noisier. So one option is that the enhanced motor noise under HFS is the cause of this decrease in error sensitivity that we observe in our data. So we tested this effect. We first measure the level of variability, the motor variability under HFS relative to control. So each dot here actually shows the variability, the motor variability, the variance of trajectories in HFS relative to control. And you can see that there is a clear enhancement of noise under HFS. But from these sets of trials, we selected a subset of sessions where the noise in control and HFS was comparable. And we computed the error sensitivity only for these subsessions where the noise level was roughly matched. And we found that even in this case, when we match the noise in control and HFS, error sensitivity is significantly decreased. Now, which may suggest that the noise the motor noise, the enhanced motor noise under HFS is not what drives the reduction in error sensitivity. Another thing that we found was that, or a way to explain how come this motor noise doesn't in itself affect the error sensitivity, is studying the adaptive response. So we used here the framework that was suggested by Tanvi and Moritz Smith. And what they showed is that in the presence of external noise, there is adaptive response. This is studies of humans, but people change in face of a large error, people try to correct in the next movement to reduce this large error. But this is only when the errors are external. As you can see here, this is just a very clearly negative relationship between the magnitude of the external error and the adaptive response. However, when the error is internal, then there is no adaptive response. So people are not sensitive to internal noise, but they are very sensitive to external noise, even when the two are the same magnitude. And what we found is both under control and under HFS, we didn't see any adaptive response. So large or small errors in HFS did not trigger any adaptive response of the monkey, which may suggest that these HFS, or the enhanced motor noise that we see under HFS, is taken by the monkey as an internal noise and doesn't trigger in itself an adaptive response. So if I summarize the behavioral results, what we found is under HFS, there is a reduction of error sensitivity and at least partially independent increased motor noise. And both of these factors affect or impair the adaptation. So we have an impaired adaptation to the force field under HFS, which is the result of reduced error sensitivity and possibly increase in the motor noise. So next we ask, what are the neural correlates of these motor adaptation? Because we have in non-human primates, we have access to activity in the motor cortex. And the question was, given the impaired adaptation, what happened in the motor cortex that could explain the impaired adaptation that we find? Now, we use the framework that was suggested by Vyas, where he actually showed that in the process of adaptation, we have a shift from motor preparation to motor generation and motor command. And the movement itself actually is important for the trial-by-trial learning process. However, what Vyas actually showed is that already the motor preparation activity, motor cortical activity during the preparatory period actually is correlated and drives the trial-by-trial learning process. Now, because we actually argue that the cerebellum, at least in the context of upper limb movements, has its main effect is mediated via the motor cortex, we now ask on which epoch or during which epoch of the task the cerebellum is important. Does the cerebellum contribute to the motor preparation or only during motor generation or motor command? So the question we ask, what happened under HFS to the evolution of activity along the motor course in the motor cortex? So this is just an example of a single cell. This is a single motor cortical cell that was recorded during the task. Now, all the trials here are aligned on the go signal. Okay, so this is a single cell. This is the time zero is the go signal. This magenta symbols shows the onset of the cue. Okay, so this is the visual cue that tells the monkey which target he will have to attend. In this case, it's always the same target. In all these trials, the monkey performed movement to one and the same target, peripheral target. This is the learned target. On the right, you can see the perturbations or the manipulations that were applied in the different trials. You can see that first there is a control trials, then there is a force field trials, washout, HFS trials, another washout, and then force field HFS trials. And these little blips in the blue line actually shows you the catch trials. So at the top, you can see the PSTH, the peristimulus time histogram that captures the activity of the cells around the go signal. Now, we can see very clearly that the HFS has a significant effect on the firing of the cells. And you can see both looking at the HFS trials here. I hope, do you see my cursor? I mean, is this, okay, excellent. So you can see the reduction of firing during HFS, when HFS is given by itself and in combination with the force field. In both cases, we see a clear effect of reduction in firing rate. You can also appreciate it here by the PSTH. You can see that the firing rate is not very different between force field and null field trials, but it's very different between control and HFS trials. But importantly, you can see that the change in firing rate starts already, that is triggered by the HFS starts already before the go signal. So already during the preparatory period, we see an effect of the HFS, of the loss of cerebellar signals in the activity of the cell. So this is example of a single cell. Now I show you the activity of 460 cells that was recorded from this monkey. You can see each line is activity of one cell in the learned target during control and during HFS. And what we ask is, are there any changes during the preparatory period under HFS that could explain the impaired adaptation in these conditions? So I will start, I'll just say that our first approach was to try and see evidence for single cell correlation, or try to correlate single cell activity with the learning curve, and this brought us nowhere, basically. I mean, sadly, and I'm a great believer in single cell activity and the activity of well-identified single cells, but trying to correlate between single cells and adaptation was, we just were, it was a big mess. So we actually joined the team of people who are studying the coordinated activity in the motor cortex and try to see whether the coordinated motor cortical activity could explain the adaptation deficits exhibited by the monkey. So the first thing, we took the activity of all the cells that we had during the control 8, what we call, that is the control trials where the monkey performed movements to 8 different targets, and asked what is the target related information in these in the activity of single cells. And what I show you here is the first, we did a dimensionalized reduction. We looked at the first 3 PCs, and I show you here the activity or the average activity of in the different directions. And what we look here is the activity from the cue here at the center to the go signal. So the go signals are these dots at the end of these trajectories. And what we see is already between, before the go signal, between cue onset and the go signal, activity, neuron activity is strongly correlated with the subsequent direction of movement. Now, this is just, this nothing new here. People have shown this. This is just sort of a sanity check. But then we ask, okay, what is the effect of HFS on adaptation-related activity? So here, I'll show you first how HFS affect activity of motor cortical or population of motor cortical cells during the movement. So this is, we looked at movement-related activity. In this case, we don't have different targets because every cell was recorded when the monkey performed all the trials to a single learned target. So we average activity across all targets. But we look at a different condition separately. What you see here is basically the projection in the first, in 3 dimensions. Okay, so this is PC 1, 2, and 3, and I just show you this rotated in different ways to hopefully make it a little bit clearer. And we can see here that there is the activity is very sensitive to the different conditions. And the conditions are control or force field in blue. Here, this is control, and this is force field, or HFS and force field HFS. So you can see all the trajectories are very well separated. But in a way, it seems that these 2 perturbations, the force field and HFS, are almost orthogonal to each other. So the trajectories, I mean, the differences between the, so there is, so if I try to say it more simply, I mean, there is effect of force field moving from here to here, and effect of HFS moving from here to here. And these 2 effects are orthogonal to each other. So, and this is what we found during the movement. But during preparation for movement, the situation was a bit different. So again, these are the 4 conditions, the neural trajectories in the 4 conditions, but now during preparatory period. And you can see that in this case, there are differences between HFS and no HFS trials. So in the absence of HFS, control and force field, the trajectories are very similar. However, in the presence of HFS, control HFS and force field HFS are very different. So for some reason, under HFS, the differences between or the effect of force field is much larger than in the absence of HFS. And you can see this also when we try to classify. So if I take a trial and try to say whether in this trial we will have force field or not, or whether this trial is coming from a force field conditions or not, the accuracy of this classification is much better under HFS than under control. Now, obviously, we can still do very well also during control conditions, but in HFS, the separation between force field and non-field trials is better. And this is interesting because we are all talking about here, this is a preparatory activity, which means there is no force field still. So next we ask, before we ask, I mean, during HFS, preparatory activity changes and becomes more affected by the subsequent force field. So this is on average. And the next point that I'll try to make is, or I'll try to ask, is whether these changes in the preparatory activity under HFS or in the absence of HFS are correlated with the adaptation process. So here again, we were looking, so we used the framework that was suggested by this paper of Vyas. And what he did, he looked at the activity. So this is basically the target-related dimension or subspace of neural activity. And each of these large dots actually represents the preparatory activity for a given target located in space. But we can also project single trial activity on this subspace. And what Vyas have shown is that the single trial activity, or slowly, and this is in under visual motor rotation, he showed that the single trial activity actually rotates in visual motor rotation task along the delay period, such that the neural angle during the delay period gradually shifts towards the optimal angle that is required for performing this visual motor rotation task. Now, we used the same approach, but for the force field manipulation. We first identified the target-related subspace, and then we looked at the activity of single trials along the adaptation process and tried to see whether there is any change in the neural angle that could be measured here, the activity of the single trials relative to the learned target as a function of adaptation. So we use this angle, this neural angle relative to the learned target as a measure for adaptation. And we ask, is there any correlation between the neural angle and the adaptation process? And this is what I show here. And here we measure the neural angle from the learned target, and I showed basically the movement error as a function of this neural angle. And light dots actually shows early trials, and dark dots show late trials. And we can see that during adaptation, as the movement error decreases in force field trials without HFS, the neural angle becomes smaller and concentrates around low values, which is, it becomes very similar, or there is some alignment of the neural angle with the learned target. However, for HFS, we see that with adaptations, the neural angle actually increases and it settles around high values. In this case, almost 45, almost 50 degrees. We can see it also when we plot the angle, the neural angle, its deviation from the learned target as a function of trial number and trial number along the adaptation. And again, you can see that very fast in control or in force field conditions, the neural angle is around 0. But during HFS, this neural angle gradually increases. And this is just quantification, the neural angle at the first 4 trials and at the last 4 trials. And you can see that there is a clear increase of the neural angle under HFS, but not in the absence of HFS. What is interesting is that this neural angle was also sensitive to the direction of the force field. So such that if in counterclockwise and clockwise, the neural angle was opposite. So here again, we have differences between force field and force field HFS. And what we found is that during learning under HFS, there is a larger shift in population activity in the target-related subspace compared to force field trials. And yet, motor performances are impaired. So I will summarize. What we found is that force field adaptation is impaired under high-frequency stimulation, when cerebellar signals are blocked, force field adaptation is impaired. And we found that this impairment is accompanied by reduced error sensitivity and increase of internal noise. The neural correlate is that of this impaired adaptation is that neural activity changes both during motor preparation and execution. In both cases, the neural activity changes significantly. But importantly, under HFS, the differences in preparatory activity during force field and null field are attenuated. So under HFS, the neural activity is changing more extreme, or the change in neural activity is larger during the preparatory activity compared to non-HFS trust. And still, even though the activity of the cell become more sensitive to the subsequent force, the performance of the monkey are impaired. So for me, this is a puzzle, because I mean, if neural activity is more sensitive to the force field, how come the performance is impaired? So I was thinking about two possible interpretations, and this is a hand-waving alert. So this is just a hand-waving interpretation. I don't have any good model yet for this. One option is that cortical preparatory activity contributes only partially to the adaptation process. So the adaptation-related changes that we see during the preparation only account for partial of the adaptation process. And we still need the cerebellar translation of this motor preparation to motor command. And this translation is responsible for finalizing the adaptive response. In this case, in the absence of cerebellar signals, preparation is unaffected in theory, but the translation of this preparation into a well-adapted motor output is incomplete, and that's why performance is poor. An alternative explanation is that during motor preparation, part of the force field adaptation takes place in an orthogonal subspace to the target-related plane. And this orthogonal preparatory signal is driven by cerebellar input. So these are two different models. But in both cases, the observed learning-related changes, or what we argue, at least I won't even say argue, what we suggest is that the learning-related changes in the preparatory activity under HFS is in fact a partially effective compensatory mechanism for the cerebellar block. The monkey cannot rely anymore on the cerebellum to complete or to take part in this adaptive process. And motor cortical network needs to compensate for this partial loss. And we see this compensation by these changes in preparatory activity in the motor cortex when the cerebellar signals are blocked. So I'll just say that in the absence, I'll add to the summary that in the absence of cerebellar signals, the motor system changes the preparatory strategy, possibly to compensate for the loss of the cerebellar controller. So as I promised, I think to start with, this is still an ongoing research, and I think I'll stop here, and I'm happy to get any insight or questions or any ideas of possible interpretation of this result.
Yifat, thank you so much. That was wonderful. May I start the questions by asking you the following? In your paradigm, you have force field plus high frequency stimulation, and it's possible that the high frequency stimulation is preventing cerebellar output from reaching the motor cortex, but the cerebellum is still learning, right? So if that's the case, I wonder, you follow a force field plus HFS with washout, and I'm wondering, are the after effects in the washout trials that follow the end of high frequency stimulation any indication that the cerebellar learning was taking place, but preventing its output from being expressed during the movements?
Yeah, that's a great question that we are we're trying to address. But one, I mean, ideally, I would love to have a catch twice, HFS catch twice in the middle of adaptation. But the problem is that the HFS takes several seconds in itself to become effective. So it's like we found that it takes 3 to 4 seconds for the full block to occur, which makes it impossible for us to use catch twice. For the washout, so we have not checked it yet. I mean, so I don't have a direct answer to your question. But what I'm thinking is, can the cerebellum learn even if there is the feedback? I mean, if the motor cortex doesn't learn, what is the signal that will drive the cerebellar learning? So, you know, so for example, let's take the extreme case. The monkey doesn't get better because it's such a noisy situation that the monkey doesn't get better. Will the cerebellum learn anyway? I mean, this is a question that I have because I'm so out of my element here. I'd be happy to hear your take on that.
Vincenzo has his hand up. I don't have an answer either. Vincenzo, go ahead.
Hi, very nice talk. My questions are about behavior controls. And one is if you have tested if the HFS has any impact on behavior, like the monkey start to shake or something like this. And the second is on behavior during the preparatory activity. We saw that there is cortical activity before the go. But what the monkey does before the go? It's possible they try to push or try to move in one direction before the go.
Yeah, so we don't see this. I mean, we have also EMG, we record EMG activity. The monkey is basically attached. I mean, there is an exoskeleton. So any type of movement that during the cue, we will see it. I mean, so, and we don't see anything dramatic during the preparatory activity. Yeah, so I don't see any drastic changes in the behavior of the monkey during this during the preparation. We also have some videos of the monkey making movements under HFS and under control, and it's not very, I mean, in a very superficial manner. I cannot say that looking at the video I could say like before movement start. Once movement starts, you can see that there is something wrong, but before movement start, it's difficult to say, yes, the monkey is, there is something behavioral different under HFS. But I mean, it's a fair point. I just don't, in the EMG, in the EMG and in the kinematics during the preparatory activity, we don't see anything clear. In fact, the monkey starts its movement later, so the reaction time increases. So if anything, I mean, it takes him longer to make a movement.
Okay, thank you.
Hi, Yifat, may I ask a question?
Definitely, no, sure.
So for this, you showed that even with the high frequency stimulation, these over time, these improvement, it's not as strong, but there is some negative correlation. So these improvement. Do you see the dynamics of the population changing over time to get closer to the, to the regular force field? In case of force field, to HFS, turning towards a non-HFS activity.
So you mean in the absence of HFS or in the presence of HFS?
In the presence, but over time, because you do see some improvement. It's not, as you showed, it's not strong, but there is.
So if I go back, do you mean here, like whether there is an improvement?
I'm sorry, no, you showed me. I mean, maybe I got it wrong, but is there an improvement over time
In the behavior?
In the behavior, yeah.
Well, I mean, the monkey does manage on average. The monkey. Okay, so yeah, I want the monkey managed to have to get some improvement. What we found is that the fast improvement that occurred in the first 5 trials is less sensitive to HFS compared to the slow improvement that occurs in later trials. And in later trials, I don't think we found anything gradual. I mean, I thought that for the most part, I mean, the monkey just, got that much better and that's it. Now, the thing is that normally there is always a trade-off between how many trials you can have in each conditions and how many conditions you can have. Now, because we needed so many conditions, the number of trials per sessions was no longer than, I think, 60 trials max. So I don't know much. I mean, it would be interesting, of course, to see, okay, now to have 200 trials the monkey is doing and see whether the monkey just got like stuck on some level of okay, that's as good as I can, and there is no point in being better. That's another thing, right? I mean, if the monkey can perform, and he's not perfect, but can perform well enough, maybe he just doesn't invest the effort of getting any better as long as he gets the reward, right? I mean, it's not, I mean, that's another option, but I don't know what happened in 200 after like 100 tries or 200 tries. Yeah, that's kind of, yeah, that's the trade-off of not using human.
Okay, thanks.
Yeah.
We had a question that was placed on the chat. Maria asked the question. Maria, do you want to ask it yourself? Are you available, or do you want me to read it?
I can, yeah, thank you. I can just say it. I'm not sure that I formulate properly, but I wonder, is it possible to kind of extract or to find a task where you can differentiate the importance of the motor cortex, like, or bottom-up connections versus connections to the brainstem and or spinal cord? Yeah, so whether it can be done if we use other tasks, or yeah, what may be your suggestions about that?
Yeah, that's a good question. I just want to say that we looked in the way we started with the entire idea of high frequency stimulation, or in generally putting electrode in the SCP, is because we were looking for the cerebellar rubrospinal pathway. So we recorded in the spinal cord and stimulated in the peduncle and looked for neurons, spinal neurons that integrate this cerebellar information. And this is a small, so it is not like this pathway. I mean, it was very, very, very difficult to find cells in the spinal cord that are activated, unlike in the motor cortex, where we have like, I don't know, like 40% of the cells that receive input. So yeah, the pathway is different, it's like it is mostly going upstream. As for a task that will encourage more spinal response and less cortical contribution, I don't know, maybe some faster task. I mean, with like reaction time type of task, or task where the monkey needs to be as fast as possible without preparation, and so this kind
which may involve particular spinal, probably, like this fast.
Yeah. Yeah, I don't know. I mean, I don't. The cerebellar rubrospinal is kind of a tricky thing to. I don't know whether it's possible. Trying to think distal versus proximal, whether there is a difference, but I don't know. Yeah, so one thing is to. Yeah, no, I don't have any good idea of like. Yeah. Yeah, unfortunately, I don't have a good idea of a task that will obliterate, like, or will not need cortical processing, and will emphasize the spinal circuitry.
Yeah, I see. Yeah, just we are trying to understand how much could we stimulate cerebellum using like transcranial magnetic stimulation, and this is a big issue where we stimulate and what are the tracks we do stimulate, but with the involvement of the reticulospinal tract is a big issue, because like all this, the only thing you can sometimes find at the EMG is supposed to be from the reticulospinal tract, but
Yes,
I was wondering if it's possible to track it in animal studies, like to involve this track specifically.
Wow, that's complicated. Yeah, these are like, yeah, I have to think about it. I don't have any, I tend to forget, for years I worked on the cortical spinal tract, but now I'm trying to forget about this pathway, and definitely the reticulospinal, which is even more complicated. But yeah, I don't have a good answer here.
Thank you so much.
Yifat, Dan has a question.
Yes.
Yifat, that was a beautiful talk. A lot of things to think about. I'm trying to think through how the when you showed the slide with the increased angular deviation during HFS, where you have the beautiful neural state curves. This one.
This one.
Yeah, that one. Yep. I just wanted to make sure that I'm understanding this correctly. So this is, so in the second plot from the left, this is, you're basically, the preparatory state starts from sort of its usual state, and then it starts, as we said, rotating towards in the direction of the adaptation, or consistent with compensating for the adaptation. And then in high frequency stimulation, it just keeps going. And at least within the first, this many trials doesn't saturate.
Yeah.
Is it, and then I think the last panel you were saying it's the opposite, it rotates in the opposite direction. So this is consistent across different sessions or different learned targets? Like, is it really that it continues rotating, or is it just that there's, in this particular session, it went towards a point that happened to be passed?
This is average across all sessions in this monkey. So in this specific monkey, we just we computed it across sessions and average according to the trial number. The way I view it is, in a way, normally when the monkey is just trying to prepare for like a movement in the force field. So the monkey is just still planning the movement to the learned target, and he doesn't. It's not like in the visual motor rotation. He doesn't plan going leftwards or rightward or any other direction. He still, after he adapts, he's using other mechanism to compensate for this perturbing force, but the plan is to the same target. However, in the HFS, at least in this case, and we still have to, I will afterwards say what is the possible caveat. At least in this case, the monkey is just possibly planning. It cannot use any internal model, any prediction to compensate for the force. So he's either using the force field he say, okay, it doesn't say, but it's like he's planning to nearby target or to a different target, in the hope that the force field will push it to the right, because the mechanism is just not there for, I will plan this and some magic will compensate for the force, because this magic is gone, in a way. This is a very, hand waving, again, it's a hand waving, and it is possible, and an alternative, that it's not that he's planning something different, but the plan is just, he just doesn't plan to the right target, because he has no a good model for how my plan will turn into movement. So it's not that he's planning rightwards or leftwards, he's just planning, the plan is corrupted, and there is some noise. And in this case, it was there is some consistency with maybe re-aiming, but yeah, that's the way I envision it. Is this answering your question?
Yeah, no, I just, it's such a profound effect in this plot. I just wanted to make sure I understood it. And yeah, no, it does. I guess maybe one quick follow-up. Do you see, if you look at like initial takeoff angles, so the initial direction that it starts reaching it, is that consistent with the difference in this angle neurally, or are they disconnected?
No, that's the next thing we should do. Really look at the early. The question is how early. I mean, but yes, we definitely should do it. I also want to say that we have the same data with when the monkey is performing the task without visual feedback, and I think this would be interesting to compare with and without visual feedback how is actually performing this under HFS. But yeah, I'm like it's always the plan for tomorrow, but we still didn't get there. I mean, but that is the, yeah.
Thank you. This is fascinating.
Thank you.
Pat, thank you so much. That was wonderful. It was a pleasure to having you, and I look forward to hearing how these analyses that were suggested turn out. It's really intriguing stuff.
Yeah, thank you so much. This is really great. I mean, it's a pleasure to get all these input from people that really know the learning and cerebellum. It's like I feel very privileged.
Thank you so much, Yifat. Everybody have a good Tuesday. Bye-bye.